Thank you very much, Max, and very pleased to be there and to see you again. So I will start this talk. This talk about division of effects resulting from network geometry in mixed treatment comparisons. And we will discuss meta-analysis about major depressive disorder. I must say that the person who did the analysis is Constant. Constant is here today, but I think he was a little bit too shy to discuss it, so I will talk. But actually, a lot of the works that I will present were done by Constant. So kudos, Constant. And I have to disclose a few things. So first thing, I am a former postdoctoral at Metrics. So I heard about your call, Max, about the postdoctoral fellowship. And I will say that it's a super opportunity. Actually, it changed a lot of things for me. And I would love to be again a postdoctoral fellow. And the second conflict of interest is that we work together for long, Max, so I know the moderator, so actually he will be kind with me, hopefully. And I don't have any specific conflict of interest with pharmaceutical firms, and I get my fundings from various public funds in France and Europe. I will talk to you about a topic that was of interest to me for long, because it was a topic related with my medical and my PhD, actually, and my medical thesis. And when I started to work in this area, there was this very important meta-analysis about antidepressants. It was a network meta-analysis that tried to rank the various antidepressants we are used to use in our routine practice. So when I started, there was this stellar study, and this stellar study also that appeared in the New England Journal of Medicine that looked at publication bias for antidepressant trials. So you see all antidepressants are here. Each square is a given trial of this antidepressant against placebo. And you can see above the red line, the positive trials, and just under the red line, the negative trials. And what you can see here are the trials that are published in the literature. And here you can see the trials that were submitted to the FDA. So not necessarily you have a lot of trials that were unpublished. Actually, those were the negative studies, but you had also a lot of negative trials that were published as positive trials. So this is the literature where that I start to work on. And at this time, I had this idea. It was quite a silly idea, but I always have silly ideas, was to mix both the Cipriani study and the Turner study to discuss the issue of publication bias and limitation of knowledge. And actually, I built a meta-analysis to compare placebos, not antidepressants, but it was an indirect comparison of the various placebos of two antidepressants, fluoxetine and venlafaxine. And it was a tongue-in-cheek paper, and we discussed at the end that perhaps there is so much publication bias that we cannot even be sure that sucrose equals sucrose in major depressive disorder. So I'm saying you this because when I discussed this in my team, I had two comments. The first one was, Florian, a psychiatrist should never say that. So this is something that I heard. And as a PhD student, it's fun, but it's not always so fun. And why are you wasting so much intelligence on this? Actually, today I'm super happy because I will try to answer both questions or both things, but those are my starting points. Actually, why to waste so much intelligence on this? I would say that I didn't waste a lot of intelligence on this, or at least I didn't wasted a lot of time, because I had some data and I reused data for this specific meta-analysis. Actually, some colleagues wasted much more time on that question, because they use much more data to compare all placebos of all antidepressants. And guess what? They found some differences between placebos. So they discussed that this is likely due to bias, and of course, I agree that it's likely due to bias. And they say that this bias could be because of unblinding or so on. But actually, it could be also by a lot of things that are difficult to handle in network meta-analysis, such as the similarity assumption between trials, problems of inconsistencies that can be actually that can lead to this kind of results that are not expected results. And there is this super paper in Science that discusses the meta wars, and that says everything in the sentence. Meta-analyses are supposed to end scientific debates, but sometimes they can cause more controversy. And for instance, if we look at this meta-analysis for antidepressants, that found that one of the drugs, the most important, was escitalopram. Actually, if we look at this study, this network meta-analysis, a few years later, we had a second network meta-analysis that found that there was no big difference between antidepressants, so quite the opposite. And as a clinician, it's problematic because meta-analysis are supposed to be like references and exhaustive and reproducible review of all existing and available evidence, but in some occasions, they are not reproducible at all. So then I came in Matrix, and with John and also Clément, who is also involved in the new study, we decided to try to explore this issue of reproducibility in meta-analysis by applying the concept of vibration of effect. So the concept that John published with Chirag Patel, it was in epidemiology research. So we try to use it in the field of meta-analysis. And we started by a case study comparing two treatments, nalmefene and naltrexone, that we give for alcohol use disorder. And you will see we made a lot of possible choice, like the choice regarding the PICOS of the meta-analysis. So choice regarding which patient to include, which intervention to include, which comparator to use, and so on. And based on the PICO, we were able to perform more than 9,000 meta-analyses comparing nalmefene and naltrexone. So this was an indirect comparison. And we got this result. So this is the figure we find in most of the vibration of effects projects. So on the x-axis, you have effect sizes, and on the y-axis, you have p-value on a log scale. So beyond this line, you have a p-value that are smaller than 0.05 and larger here. And here you have effect sizes. Okay, so we were able to generate some meta-analysis that supported the fact that naltrexone was superior to nalmefene, meta-analysis supporting that naltrexone was inferior to nalmefene, and actually meta-analysis that found no differences. And it's pretty easy to cherry pick some in both different directions and to ask clinicians if they are, let's say, convinced by the selection criteria. And in some occasions, it can be convincing. So we made other case study, one in acupuncture. Acupuncture is super interesting because this is an intervention that has likely no biological plausibility, but has many clinical heterogeneity in the intervention. So actually, you can make a lot of choice regarding the PICOS. So we studied also vibration of effect, and we found some vibration of effect in a direct comparison between acupuncture and inactive comparators. We explored also this in the individual patient data meta-analysis context by making all possible pooled analysis, because you know that we have a lot of pooled analysis in biomedicine, and we try to adapt this. And we did this in type 2 diabetes mellitus. And actually, there was not that much vibration of effects for certain outcomes, like glycated hemoglobin. And this is expected because this is something that is very often observed in clinical trials for those diabetic drugs. We had not that much vibration of effects for major adverse cardiovascular events, perhaps because we chose the drug canagliflozin that had quite an effect on this. And we were able to identify much more vibration of effects regarding serious adverse events, depending on the studies that were included. So let's go back to our new case study, because what we did with Constant was just a new case study, but this time it was not about a direct comparison, an indirect comparison. It was not about individual patient meta-analysis, but we wanted to apply this to a much more complex network. So the networks on antidepressant research, and we have a new meta-analysis. Actually, John was an author of this meta-analysis too, and it's a massive meta-analysis about 21 antidepressants, and actually placebo in this. So here is a network. You have in this meta-analysis a specific analysis for efficacy of the drug, like the response rate, and for, we will call this acceptability, but it's rather I would say it's not directly acceptability, it's rather treatment discontinuation. So these are the two networks. And actually, we had some fun, and it took a lot of time, because we tried to make from these networks all possible networks that could be explored. So for instance, all possible networks with 2 treatments, 3 treatments, 4 treatments. So actually, we had 4 million, more than 4 million network meta-analysis to perform in this study. So I'm super happy to say that I have in front of you the world record of meta-analysis in the world. So it's Constant. He performed 4 million meta-analysis, but not once. He did this twice, once for acceptability and one for, let's say, treatment discontinuation, meaning, so efficacy, sorry, and treatment discontinuation. So we performed 8 million meta-analysis. I hope that this record will stand for long now in the medical literature. So we perform all these meta-analysis and had a look at all comparisons for vibration of effects, at all possible comparisons. So what was very challenging in this work was to perform all the comparisons, because it took at least, I would say, 3 months to perform all the analysis. So the computer were running and running and running and running. So then we got this. Here is the treatment, what we observed for clomipramine. In this, so clomipramine as compared with all other antidepressants. For instance, here, this is the comparisons between clomipramine and agomelatine, clomipramine and amitriptyline. So all the antidepressants, and actually placebo here, compared with clomipramine. I show you this one because this was the one where there was the most important vibration of effect that we identified. I will show also placebo. Placebo, it's the opposite. It's where we found the less vibration of effect. So for instance, agomelatine versus placebo, all effects were in the same direction, and actually all were statistically significant. For amitriptyline the same, bupropion the same. Perhaps clomipramine had some vibration of effects versus placebo, but not that much. And milnacipran a little bit, but not that much. But for all the other, there was no vibration of effect for placebo. So it won't be interesting if I show you this figure for all treatments. That's why I show you the one with the most vibration of effects, the less vibration of effects, and then we try to summarize all we found in these figures. So this one on the left side is for efficacy, and this one on the right side is for treatment discontinuation. You can see that placebo had the less vibration of effect, because when it's green, it's good, meaning that the center represents the fact that we found a Janus effect, meaning that the first percentile and the 99 percentiles are in opposite directions. So in green, there is no Janus effect. In orange, there is a Janus effect. And just around, it's if in case there is a Janus effect, were we able to find statistically significant results in opposite direction, meaning that depending on the network for this comparison, I can find networks that show that one drug is superior to the other and one drug is not superior to the other. And this is what happened in red. I would say it's around 25% of drugs that can have that for those drugs, I'm able to find some meta-analysis in one direction or in the other direction. Actually, vibration of effects was quite correlated between two outcomes, discontinuation and treatment response efficacy. And we perform also a few exploratory analyses to try to identify what was the driver of this vibration of effects. And without a big surprise, when you have only indirect evidence for a comparison, you have the more vibration of effects. So this increase actually the log relative odd ratios. It is related with Janus effect and with statistically significant results in opposite direction. So the fact that we have only indirect evidence when compared with direct evidence, and there is direct evidence without inconsistency, direct evidence with inconsistency, we didn't find a lot of things, and only indirect evidence, there was a signal. Then for effect size, we looked if the effect size in the biggest meta-analysis were close to 0 of the comparison. And when the effect size was close to 0, of course, there was more Janus effect. Of course, we were able to generate more statistically significant results in opposite direction, but no change in relative log odd ratio, meaning that vibration of effects, so the extent to which the point estimates might change is highly dependent on the fact you rely only on indirect evidence. And when the effect size is small, of course, it can go in both directions, meaning you have the Janus effect and so on. So actually, problems are for treatments that are not different one from each other. And this is super interesting for antidepressants because most of antidepressants are not very different one from each other in terms of efficacy. So now what is tricky with this is to interpret the results. It's not so simple. So I put this as a list of good news and bad news. So the good news, I think this is what is expected where we are doing 4 million meta-analyses. It's not surprising that we can find in some occasion, and especially when the treatments are not so different, differences between, so not differences, but meta-analyses that can say all and the opposite. So it's not so problematic, and actually we use some measure of uncertainty when we are doing meta-analysis, and this is for this reason. Of course, we relied sometimes on the term statistical significance, and we should not rely that much on this in meta-analysis. And clinicians, of course, have some understanding of uncertainty. At least we can hope. Then I would say it's only theoretical. So this is a good news. This is a case study that is only theoretical. So we just perform all these meta-analyses here in Rennes, and this is not something that happens, of course, in the literature, because on this topic, you don't have 4 million meta-analyses. Of course, the good news is that if we rely on the most exhaustive meta-analysis, we are super happy because meta-analyses are exhaustive review of all existing evidence. So there is no reason to select network geometries. Of course, it's good because meta-analyses are registered a priori, for instance, on Prospero, and little change changes happens between the protocol when the studies are published and their registration. And the other good news is that direct comparisons are protecting us from poor inferences, because at least when we have direct comparison, we have much less vibration of effects. And you can see, for instance, for placebo, there is much less vibration of effect, and there were always direct comparisons, of course, versus placebo. So those are the good news. But the bad news. One should not rely on statistical significance in meta-analysis, and clinicians have an understanding of uncertainty. I would say, are you sure? So I call this the bold effect, because if you look at the Cipriani meta-analysis, when at least it was not a confidence interval, but a credibility interval, did not include one, they put this in bold, meaning that you should look at this, because here there is a big difference. And clinicians actually, sadly, they don't have always the big understanding of uncertainty. This is only theoretical. Okay, are you sure that it is only theoretical? Actually, I presented you this comparison for nalmefene when we explored vibration of effects. Actually, for nalmefene, we did this because there was one meta-analysis performed by Naudet, the manufacturer of nalmefene, that identified actually a superiority of nalmefene versus naltrexone. And there were also other meta-analyses that found no differences. So actually, conflicting meta-analysis based on various methodological choice happens in the literature. Meta-analyses are exhaustive review of all existing evidence. Are you sure of that? So we did that also with John a few years ago. And we had these interesting results about network meta-analysis in the treatment of rheumatoid arthritis. We found that there was a lot of network meta-analysis that were in overlap on this topic, and none was exhaustive of all existing treatments. None was exhaustive of all existing references that could be included in the meta-analysis. And when I say references, I mean not all studies. And this is by far, because, for instance, the most complete network meta-analysis we identified had less than 16 studies, where actually there were more than, let's say, more than 100 studies on biologics. So most are not complete. And actually, in psoriasis, we found that they differed a lot in terms of network geometries between the treatments that are chosen, but much more differences in terms of network geometry. For instance, how people lump treatment together by a therapeutic class, or by some like the add-on therapeutics you can have with a drug. And actually, it happens. And for this comparison, adalimumab versus etanercept, you have all those meta-analyses that are existing. Most found no effects. Some found an effect. Some found an effect in the opposite direction. And actually, it can be problematic for clinicians who have to cope with all this conflicting evidence. For instance, this is what we found for the use of biologics in psoriasis. Now, you can see that each line is a given network meta-analysis comparing various biologics. Here you have all the drugs that could be included in the meta-analysis. And you can see when there is a little X that these drugs was included in the meta-analysis. So most were incomplete of all treatments. And in green, you have the treatment is said to be the best in the abstract of the meta-analysis. Actually, it's not always the same. Some are seemingly good, but there are some change. from time to time. And in red, you have the drug that was manufactured by the people involved in the meta-analysis, because most of those meta-analyses are commissioned by the industry. And you can see that each and every time that the meta-analysis is performed by an industry, it's this drug that is put forward. So meta-analyses are registered a priori, and little change happen in PICOs in comparison with their registration. So I would say that a lot of meta-analyses are not registered a priori. And I would say that sometimes PICOs are changing a little bit. And I think this is the work by Max. So kudos, Max, for this. But there is change in the PICOs during the research. Direct comparisons are protecting us for poor inferences. This is a tricky question, and I will cite my favorite journal here. So my favorite journal now is Journal of Clinical Epidemiology. A lot of people like this journal, and I really like this journal because this journal uses in the title the main message, meaning that you just need to read the title to know what the study is about. And here, and the result actually. And it says, in network meta-analysis, most of the information comes from indirect evidence. And actually, this should not be underestimated when somebody is reading a network meta-analysis. In addition, I don't know if you remember, but I said that from the first Cipriani meta-analysis, they sold a lot. So escitalopram was the drug that was thought to be the best one. And then a lot of escitalopram was sold, at least in Europe here. It became a very successful drug. Actually, escitalopram is an enantiomer of citalopram, meaning that there is no big difference. And the strategy to use enantiomers is very known as a strategy of evergreening. And in this interesting paper, they saw that there was a big inconsistency issue between citalopram and escitalopram. And actually, if you look at indirect evidence, there was no difference. And there was difference in direct evidence. And if you read carefully at this paper, they are discussing why, in some occasion, and especially in this one, indirect evidence could be also less biased than direct evidence. So all of this is quite complex to apprehend. We looked with Constant at the specific comparison between citalopram and escitalopram because it's very interesting. Both biologically, there is no plausibility for a big difference between both drugs. And then it was very interesting because there was this inconsistency issue, and we look at vibration of effect. So in this specific comparison, there is no Janus effect. We found consistently an effect, point estimates that favored escitalopram. But when we include indirect evidence, this effect that is identified in direct comparison might disappear. And here I put two different networks for you to see that can identify a difference in favor of escitalopram or no difference in favor of escitalopram. But the bad news, I think this is what I would like to discuss with all of you today, is the fact that the vibration of effect could be used as a massive destruction weapon to say that there is uncertainty for any kind of inferences in any kind of place in medicine. And I think we need, and we are going to start to work on a small paper about that. And we need to think about that and how to use it. So I think the tool might be of interest for the field of meta-analysis in order, for instance, to explore controversies when they are overlapping and meta-analyses that are in opposite directions, when there are controversies. So, for instance, in the context of the meta wars that I told you before, perhaps we have an interest here. I think I would be very interested to use the tool now, not solely in case study, but across a wide range of meta-analysis. And I think it could be very interesting to use it to explore fields in medicine that are more subject to vibration of effects. But of course, this was the answer to our placebo study by Cipriani, actually, that we should not use this kind of tools for the purpose of ideological nihilism. And let's go back to my initial question. So a psychiatrist should not say that. So I'm fine. Now I'm professor of therapeutics, not of psychiatry. And why waste so much intelligence on this? I still don't know, but I think that when a domain can't laugh at his own flaws, it's quite dead to the core. But we answered this to Cipriani, and we said that in some occasion, this kind of reduction, it's quite absurd, I would say, to make so much sensitivity analysis, but it could be useful to explore when the medical literature in itself gives us this impression of absurdity. So thank you so much, and super happy to discuss with you all those results.